Today I will be discussing polyamines allies in plant resilience against abiotic stress. Now I will discuss first with the last keyword of my topic, that is what is stress. So you might be aware that stress could be any external factors that disrupt the normal growth, development, functioning of the plant. Another question arises that how plant face these types of stress, what could be the source. Based on the type of source, these stresses could be biotic and abiotic stresses. So generally biotic stresses are caused by living organisms like pathogens. It includes bacteria, fungi, viruses, pests like insects, nematodes, and competition from weeds also comes under the category of biotic stresses. Abiotic stresses, as you know that this is generally induced by non-living sources. This could be induced by environmental like drought, salinity, extreme temperatures. And this could be of chemical nature like heavy metals, pollution, and nutrient imbalance. So how these biotic and abiotic stresses, they are relevant in context to plants. So you will be astonished to know that these two types of biotic and abiotic stresses can reduce plant productivity by 65 to 87% depending upon the type of crop. So how these biotic and abiotic stresses could hamper the yield of the plant. So in this way, we should dig into more details how plants are exposed to wide range of abiotic stresses, biotic and abiotic stresses that significantly affect their growth, development, and productivity. These abiotic stresses include drought, salinity, extreme temperature, heavy metals, toxicity, and nutrient deficiencies, and this disrupt the physiological and metabolic processes of the plants. Among these, drought and salinity are particularly very detrimental, reducing photosynthesis and water use efficiency, whereas on the other hand, biotic stresses generally results from interaction with the non-living organisms such as pathogen, insects, nematodes, and weeds. So these stresses generally compromise plant immunity, leading to very significant loss of yield. But sometimes what happens, the combined impact of biotic and abiotic stresses could be additive, synergistic, and these leads to further damage of the crop. It means that at a time plant could face both biotic and abiotic stresses. It's generally not very common. Sometimes what happens that plants only not face biotic stresses but also abiotic stresses. So what would be the repercussion of these biotic and abiotic stresses? So this could be additive, means 1 plus 1 is equal to 2. This could be synergistic, 1 plus 1 is equal to 3, and so on. So if it is the impact of biotic and abiotic stresses becomes synergistics, it's very fatal to the plant productivity. Now come to in nutshell, I will just outline what are these, how these abiotic stresses impact the crop plants. So first I will take up salinity stress. Generally disrupts the osmotic balance by reducing water availability and causing iron toxicity. So how plant responds to salinity stress? Generally plant respond to salinity stress by synthesizing osmolytes like proline, glycine, betaine, and activate ion transporters to maintain cellular homeostasis. Then another stress is drought stress, generally reduces the availability of water, decreases photosynthesis, and generally uh, drought stress leads to closure of stomata, rolling of their leaves in just sort of conserving water. Uh, Another stress is heat stress. Generally, once plant face heat stress, generally heat stress accelerates rate of transpiration and loss of water. Whereas transpiration is triggered, so plants generally counter these heat stress by synthesizing specific heat shock proteins and acclimate themselves by synthesizing these specific proteins. Now, plants also face chilling stress, and chilling effects stress affect fluidity of membrane, activity of enzymes, causing formation of ice crystals, which damage or affect the ascent of sap, then leading to metabolic imbalance. And high light intensity generally causes photoinhibition, oxidative damage to the plants. So plants also respond to these abiotic stresses by synthesizing antioxidants and adjust arrangement of chloroplast. Whereas you see that heavy metal toxicity generally disrupts the enzyme activity and generate reactive oxygen species, triggering plants to produce metal chelators like phytochelatins and further enhance antioxidant defense system. You see that plant also face submergence stress during flooding. So what happens during submergence? Once plants are submerged, they don't get proper oxygen and face condition of hypoxia, means less oxygen for for respiration. So you see that plant adapt themselves by developing parenchyma, activating aerobic metabolic pathways. So these adaptive mechanism generally adopted by plants to cope with these abiotic stresses. And the way plant overcome these stresses is very interesting to know that how plant overcome to the hostile environment. You see this, see the photograph on one side you see that there is a drought affected wheat crop, and how severely drought affects the wheat crop is evident from this slide. On the right side you see that tomato crop is affected by drought stress. See the devastation made by the drought stress on tomato crop. So this is just mere two examples I am showing you that how drought stress affect wheat and tomato crop. Now come to that ROS, reactive oxygen species dynamics. Generally, reactive oxygen species are very central to the plant response to abiotic stresses such as climate driven stresses like flooding, freezing, ozone, ultraviolet radiation. This could be also triggered by anthropogenic processes like drought, heavy metal, salinity, sorry, pesticide, herbicides, and soil driven stresses like salinity and change of pH. So what is the fate of production of reactive oxygen species is very crucial. So you see that under normal condition reactive oxygen species are generally produced during normal metabolic reactions as a byproduct, and these reactive oxygen species play a very significant role as a signaling molecule and control growth and development of plants. So this is the case under normal condition when ROS are produced. But what happens during abiotic stresses, excessive accumulation of reactive oxygen species disrupts cellular redox homeostasis, leading to oxidative stress that damage lipids, proteins, and nucleic acid. So in one way, once reactive oxygen species are produced under normal condition, what happens? It acts as a signaling molecule. Whereas under abiotic stress condition, when there is excessive accumulation of ROS, it damages lipids, proteins. So you see that how reactive oxygen species are produced in chloroplast. Generally, during various abiotic stresses like salinity, drought, it leads to accumulation of RS in chloroplast during photosynthesis. So what happens during excessive light generally generates singlet oxygen species and superoxide radicals in photosystem first, and second, then superoxide dismutase generally converts superoxide radicals into hydrogen peroxide, which acts both as a signaling molecule and a potential oxidant. Now, what is the fate of reactive oxygen species in mitochondria? In mitochondria, generally what happens during various abiotic stresses like salinity or temperature extremes, ROS generally form during the mitochondrial electron transport chain, and generally mitochondrial production of reactive oxygen species is very much closely linked with the metabolism and stress signaling pathway. Now you see that what happens in peroxisome during abiotic stresses like drought and highlight, photorespiration in peroxisome is generally triggered and leading to increased production of hydrogen peroxide by glycolate oxalate. Peroxisomal reactive oxygen species are very much critical for metabolic adjustment during stress. Now what happens in apoplast? So during salinity and drought and heavy metal stress activates NADPH oxidase in apoplast, and the increased activity of NADPH oxidase generally generates superoxide, which is then converted into hydrogen peroxide by superoxide dismutase. So generally accumulation of ROS in apoplast contribute to cell wall modification, and you see that stress signaling and defense responses. So other organelles like endoplasmic reticulum also produces reactive oxygen species during folding of proteins, whereas nucleus receives reactive oxygen species signals. Generally these are perceived by different genes, specific genes. So the way plant generally cope with these excessive production of reactive oxygen species. So what is the fate of these reactive oxygen species, how they are changing different metabolic pathways in the plants? So generally reactive oxygen species serves as a secondary messenger in stress signaling pathways, activating transcription factors and genes involved in stress tolerance. So this is the dual role of reactive oxygen species, which highlight the complex and vital functions in abiotic stress adaptations. Now you see that in this slide, when plants are exposed to high light stress, what is the fate of photosystem 1 and 2? So once plants are exposed to high light stress, the oxygen evolving complex in chloroplast generally absorb excess light, which damages the oxygen evolving complex and the photosystem 2 reaction center. Then it breaks down important proteins also like D1 and D2 protein, leading to photoinhibition. What is photoinhibition? That is slow down of the photosynthesis. And abiotic stresses like drought and salinity further aggravate this problem by producing the reactive oxygen species. So generally this disturbs the balance inside the chloroplast, damaging reaction centers and blocking their repairs. So this further aggravates the problem, and these stresses also harms the structure of chloroplast, causing it to shrink or swell, breaking down its layer and pigment protein complex like chlorophyll. So as a result, photosynthesis slows down, and you see that these stresses also interfere with the light reaction in photosynthesis. They block the electron transfer, stop enzymes from working and reduce production of ATP, ultimately halting the plant's ability to make food. So what is the why abiotic stress tolerance is crucial for plants? So this is a very important challenge. You will be surprised to know the data that impact of drought stress on production of certain crops. I will just share that by 2050, the global population is expected to reach 9.9 billion, creating a, you see that massive demand for food. So to meet this burgeoning population, the food production needs to increase by 60% as compared to today. And currently crops like rice, wheat, maize and soya are only increasing at the rate of 1.6% per year. So to meet the future needs, the growth rate must be at least 2.4% annually. Like major crops like rice, which feeds about more than 3.5 billion people, is growing just at 1% annually. And you see that these four staple crops provide about 2/3 of the world's calories, making their productivity is very crucial. So climate change and outdated agriculture practices are making or you can say worsening this problem more. So by 2050, we will need to double grain production from 2 billion to 4 billion tons annually to feed our population. So to overcome these challenges, how these abiotic stresses challenges could be overcome, various omics approaches are essential for studying plant response to abiotic stresses and improving agronomic traits like stress tolerance, like plant height, grain quality and yield. So genomic studies generally focuses the entire DNA of a plant to identify genes linked to abiotic stress tolerance. Various techniques like quantitative trait loci mapping, genome-wide association studies and next generation genome sequencing generally address these stress-related genes, and these tools provide further insight into traits like drought tolerance to salinity, heat and other stresses. So genomics approach also supports modern breeding methods like marker assisted selection to develop stress resilient crop. Metabolomics is very much significant, which has been magnificently applied to plants to detect novel metabolites, genes and their metabolic pathways. This is my current thrust thrust area where I am working nowadays. So you see that metabolics approach is very crucial. All of you just pay attention to this approach because this is very crucial to address how metabolite profile changes in response to various abiotic stresses. So metabolites produced by plants which play a crucial role in stress tolerance or stress responses. By analyzing changes in metabolites like sugars, amino acids, and organic acids under stress condition, generally biochemical pathways are changes during various stress adaptation. For example, accumulation of osmolytes like proline and glycine betaine helps plant to retain water during drought stress. So metabolomics also reveal that how plants manage energy production and detoxify harmful substances under stresses. So these informations could be very much helpful for plant breeders and engineer crops with enhanced stress tolerance by targeting key metabolic pathways. So abiotic stresses significantly affect plant growth and reduces plant productivity. And so to combat these challenges, plants generally use signaling molecules such as phytohormones, and these natural regulators can mediate stress tolerance by triggering various adaptive responses. Phytohormones like abscisic acid, auxins, gibberellins help plants to adjust to these abiotic stresses. For example, ABA helps to reduce water loss during drought stress by closing stomata, whereas auxin and cytokinin regulate growth and stress-induced expression of genes. So by leveraging phytohormones, we can develop strategies to improve plant resilience and enhance crop production under stressful condition. So phytohormones are basically very much essential regulators that help plant to adapt and survive under abiotic stress condition by activating various protective mechanisms. So out of these phytohormones that are employed to cope these challenges, I will only confine to polyamines. So polyamines, they are different amino group like putrescine having is a diamine, spermidine, spermine. These play a very crucial role in regulating plant growth and various stress responses. So polyamines basically protect plants from oxidative damage, stabilize cell membranes, and promote root growth. So these polyamines also improve photosynthetic efficiency by protecting chloroplast by enhancing pigment synthesis. So you see that polyamines, melatonin, brasnoistride, salicylic acid, say these they work together to promote key physiological processes in plants. So cumulatively, their effect could be additive, synergistic, and all these molecules like melatonin, brasnoistride, salicylic acid, and polyamines, they improve gas exchange parameters, enhance pigment content, and regulate root architecture for better water and nutrient uptake, and increase develop enzymatic system of the plant to cope with these challenges. Additionally, these molecules also help in reducing heavy metal uptake, supporting the survival of the plant under challenging environmental conditions. So I am going to discuss in more detail regarding the role of polyamines in plants. So just you see that these polyamines, they are found in living organisms, both in plants and animals, and even in some viruses. in plants, generally these polyamines, they are present in free form, and the most common polyamines in plants are putrescine, spermine and cadaverine. Uh, but you see that these polyamines, they are distributed differentially across the plant tissues, organs, and at different growth stages. For example, you see in tobacco, uh, putrescine and spermine are more abundant in young leaves and shoot tips, whereas spermidine is higher in older leaves. So higher polyamines like spermidine and spermine are mostly produced in aerial parts of the plant like shoot and apical meristem, whereas putrescine is mainly produced in underground parts such as roots. So among these main polyamines, uh, generally spermine and spermidine are more evenly spread throughout the plants than putrescine. Uh, Polyamines generally also accumulate in different parts of the plant cells, like in carrot cells. For example, putrescine is mainly found in cytoplasm, whereas spermidine in the cell wall. So you see how there is a differential accumulation of polyamines in plants. Now, this is the image retracted from one of my recently published paper in chemosphere. So you see that here polyamines biosynthesis generally you see that begins with the decarboxylation of amino acid ornithine or arginine, and thereby producing putrescine is a simple diamine. So you see in this figure that this process could happen through two pathways. One is known as ADC pathway and another one is known as ODC pathway. So polyamines could be synthesized by adapting these two basic pathway. One is ADC pathway, another one is ODC pathway. So several enzymes work together to produce putrescine. After that, spermidine and spermine are created by adding aminopropyl groups to putrescine with the help of enzyme like spermidine synthase and spermine synthase. So the aminopropyl groups comes from methionine, which is processed by SAM decarboxylase. Under these conditions, what happens? Plant regulate generally level of polyamine through catabolic pathways. So you see that biosynthesis and catabolic pathway run in a balanced condition to maintain proper homeostasis of polyamines. So these pathways generally involves enzymes such as DO and PO, which breaks down the polyamine and produces byproducts like pyrroline, ammonia and hydrogen peroxide. So in catabolism of polyamines, you see that one of the reactive oxygen species, one of the antioxidant is produced, namely hydrogen peroxide. So you see that the accumulation of hydrogen peroxide can cause damage to various lipids membranes, and it also plays very important role in strengthening the cell wall of the plants, which helps to confer tolerance to the plants. So the level of polyamine in plant cells are generally very much tightly regulated through their synthesis or breakdown, ensuring that the plants has the right balance of polyamines. So this balance is basically very much essential for plant growth and ability to cope with the challenging environment. And generally polyamine homeostasis is maintained by a complex feedback systems that control the production, breakdown and movement in and out of the cells. So in addition to this, polyamines are also involved in producing their other biologically active compounds and support plant health and confer stress tolerance. So you see that polyamines, they are involved in regulating various physiological processes of the plants like growth and development, where they regulate cell division, elongation, differentiation, support the root and shoot growth, flower development and fruit ripening. And in addition to this, polyamines also regulate various abiotic stress responses like just I mentioned drought, salinity, and these polyamines basically stabilize cell membranes, scavenge reactive oxygen species, and enhance antioxidant defense activity to protect plant from damage. So these polyamines could be also involved in mediating defense responses like polyamines contribute to plant immunity by strengthening the cell walls through lignification, triggering defense related signaling pathways and producing reactive oxygen species to combat pathogen attack. So how these polyamines help in to delay senescence? So generally polyamines delay senescence and aging by reducing oxidative damage, by maintaining proper chlorophyll content and protecting cellular homeostasis for further adaptability to the plants. Now this slide shows the impact of polyamines in different plant species grown under heavy metal stress. Various case studies have been cited in this table regarding the role of polyamines in heavy metal stresses like cadmium, chromium, lead and aluminum on different crops like wheat, rice and vigna. So just to have an overview of how polyamines modulate different abiotic stress in plant cell in a nutshell, you can just have an overview through this table. Now, role of polyamines in further in stress tolerance, how polyamines mitigate various stresses like salinity, water and heavy metal stress. They are further summarized in this table. Now just I will take a few case studies done from my lab. So what is the role of polyamines like putrescine and spermidine in mitigating manganese induced toxicity in Brassica junctia? So that I will discuss. So what happens once plant faces toxicity of manganese? What happens? It leads to the development of brown speckles. You see on the brown speckles here, and generally this brown speckles develop on mature leaves. So this is the case with manganese toxicity. So the intensity of brown speckles formation generally could be used as a simple and rapid method for evaluating various cultivar for manganese tolerance. In addition to this, intraveinal chlorosis and necrosis are additional symptoms of manganese toxicity, and these symptoms may be accompanied by distortion in the young leaves known as crinkle leaf. So this crinkle leaf is one of the repercussion of manganese toxicity, which is characteristic indication of calcium deficiency also. And this manganese toxicity generally often leads to deficiency of other nutrients like calcium, magnesium, iron and zinc. So generally the induced deficiency of iron and magnesium results from inhibited uptake across the plasma membrane and competition or imbalance at different cellular levels. Additionally, this manganese toxicity can also lead to loss of apical dominance and increased formation of auxiliary shoots, and this condition is referred as witch's broom. You see in this figure witch's broom. So in this slide you see that how manganese toxicity is affected in different crop plants, how photosynthesis is modulated. So just quick overview of manganese toxicity in different crop plants could be generally redrawn, retracted from this table. So you see, this is the finding from my lab that how the polyamines modulate uh, response of plant, particularly various growth traits like length and root of shoot, length and length of root and shoot, fresh and dry mass of the root and shoot, and leaf area. And you see that all these parameters decrease in presence of higher concentration of manganese, but how application of spermidine at 1 millimole just recovered the damage induced by higher concentration of manganese. The other parameters like leaf area, spared value and photosynthetic traits, they were also considerably reduced at higher concentration of manganese, and these stress plants once treated with spermidine or putrescine, these counteracted the toxicity induced by higher concentration of manganese. So you see, this slide shows the impact of stomatal behavior under higher concentration of manganese. Generally, what happens at higher dose of manganese leads to closure of stomata, whereas you see that later on exogenous application of putrescine, spermidine or putrescine generally restored the damage induced by higher concentration of manganese. Now, this slide shows the application of spermidine or putrescine alone in stress-free plants, which generally enhance the photosynthetic efficiency of PS2 as compared to control. And generally, under unstressed conditions, spermidine and putrescine generally increased these parameters, and once spermidine and putrescine were applied on the stress plants that have already been given higher concentration of manganese that overcome the toxicity generated by manganese. Then come to the antioxidant enzymes. Antioxidant enzymes were further triggered in response to higher dose of manganese, and generally this application of putrescine or spermidine generated additive effect and on the antioxidant activity. So this is the model showing the how polyamines plant responds to polyamine and heavy metal stress. Generally, plants exposed to heavy metal stress changes the level of phytohormones, generates production of reactive oxygen species leading to oxidative stress, and further activation of defense system are triggered generally. Heavy metal stress generally affect the metabolism of polyamines, affecting both synthesis and its breakdown, that is catabolism. The activity of various enzymes like ADC and other enzymes generally increased, leading to an increased production of putrescine levels, and putrescine generally acts as a signal indicating the presence of stress, which in turn enhances the biosynthesis of other polyamines like spermidine and spermine. Basically, spermidine and spermine are primarily responsible for regulating stress responses, interacting with reactive oxygen species, phytohormones and transcription factors, and other molecules to manage the both stress responses and various essential cellular processes. Metal stress also increase polyamine catabolism through increased level of enzyme activity, producing beneficial byproducts like GABA and hydrogen peroxide. High polyamine levels generally activated oxidative system defense mechanism through production of hydrogen peroxide mediated signaling, scavenging of reactive oxygen species, interaction with other enzymes, and protection of cellular components from oxidative damage. So there is also proposed role of polyamines directly in heavy metal stress, although further research is needed to confirm this hypothesis. And this polyamines also enhance the glutathione levels, boosting the biosynthesis of phytogelatin conjugates, which generally activate heavy metal detoxification. So, what is the conclusion of this finding, that how manganese stress affect the plant? That manganese stress reduce the movement of stomata, decreased photosynthetic traits, lowered the biochemical parameters in Bresca junctia. And then what is the impact of polyamines, spermidine or putrescine application? It led to the improved stomatal movement, photosynthesis and growth rates were enhanced significantly, and antioxidant systems were improved, reducing oxidative damage. And then one significant aspect is application of polyamines decrease the accumulation of heavy metals. So if I talk about the comparative effectiveness of two polyamines, I have taken spermidine or putrescine. So out of these two polyamines, spermidine were found more effective than putrescine in mitigating manganese-induced stress and restoring plant health. So this is the role of bresnoestride and spermidine in combination in mitigating manganese stress in Bresca junctia. I will skip this. This is due to time constraint. Now, what is the role of polyamines in manganese-induced toxicity? So, you see that higher concentration of manganese supplied to Bresca junctia through soil generally reduce the root morphology, reduce the growth, distorted the root morphology and altered the stomatal behavior. And application of spermidine through foliage proved very beneficial and reduced the production of reactive oxygen species, and this was further confirmed by the microscopic studies. What was the fate of stomatal movement? Now, advanced molecular approaches such as transgenics, proteomics, metabolomics, they are also being employed to manipulate metabolism of polyamines and to study their role in stress tolerance. Now, next question arises, how polyamines metabolism could be manipulated for conferring abiotic stress tolerance. I am only focusing polyamines, how its metabolism could be manipulated. So generally, various studies have shown that particularly in Arabidopsis, that the production of polyamine is affected by different environmental stresses. So it was revealed that certain polyamine producing genes, they are turned off and they struggle more with the stress, suggesting that polyamines play an important role in helping plants to resist various sort of stresses. Additionally, genes responsible for production of polyamines from various organisms, they are further incorporated into plants, and then plant tend to show better stress tolerance and resistance. This is linked to the level of polyamines. So if I ask you what is the role of polyamines level in plants, so polyamines level is very important in plants in mitigating various abiotic stresses by regulating both its biosynthesis as well as catabolism. So, in addition to this, various stress related metabolites, they are upregulated by application of exogenous polyamines. They regulate different metabolites that enhance photosynthesis in plants like in cucumber. Also in other stresses like salt stress by regulating stomatal opening and closing. Polyamines also regulate different metabolites that help in the expression of polyamines like diamine oxidase, polyamine oxidase, NADPH oxidase, and also modulate the production of hydrogen peroxide. So, exogenous polyamines generally help plants to tolerate metal stress by regulating different metabolites and thereby protecting cell membranes by forming various complexes like phospholipids and with other metals. They boost glyoxylate system activating enzymes like GLY1 and GLY2 to detoxify harmful methyl glyoxal. So, in this way polyamines also increase the level of other metabolites such as glutathione, which help in metallocylation and transport of detoxification. They improve the activity of various enzymes just I mentioned previously like SOD, catalase, ascorbate peroxidase, and non-enzymatic antioxidant like ascorbate reducing oxidative stress. So I will skip this. So you see this table shows the list of metabolites differentially accumulated in tomato leaves and how various polyamines alter the metabolic profile in tomato plants. So you see that through these whatever the metabolite is expressed in higher concentration that could be sorted, and then a specific pathway could be drawn that how these pathways are altered and which intermediate is highly expressed, and accordingly the enzymes could be identified so that the problem could be correlated that how polyamines altered the various metabolic pathways in the plants. So, by identification of wide range of differentially expressed proteins and their further exploration of molecular mechanism also reveal more detail about how polyamines regulate various metabolic pathways. Now I would conclude that high polyamine content for short-term stress is beneficial for the plant, and elevated polyamines level either through exogenous supply or through genetic manipulation confer tolerance to short-term exposure to the plants grown under various abiotic stress conditions. In addition to this, application of spermidine or putrescine or both to stressed and stress-free plants modified the movement of stomata, improved photosynthesis and growth rates, and also enhanced antioxidant system, whereas accumulation of manganese and production of superoxide radicals were reduced. And out of, if I consider out of these two polyamines which I had discussed like spermidine or putrescine, spermidine was found more significant, prominent in restoring the damage caused by manganese. So, what are the future challenges? That unraveling various metabolic pathways with regulatory nodes by adopting metabolic approaches will going to help to identify unique or shared regulatory nodes within metabolic pathways affected by polyamine metabolism, which will further decipher details and crosstalk among different pathways that influence various abiotic stresses. Then, in addition to this, targeted genetic engineering for conferring stress tolerance is further needs to be addressed by identifying the key regulatory targets in metabolism of polyamines will further reveal the development of genetically engineered plants to confer tolerance under abiotic stresses. Then there is a need to optimize regulation of polyamines for various stress responses. So this could be the regulation polyamines level in the plants could be controlled by controlling various the specific metabolism related to polyamines in a controlled manner or tissue specific manner by activated early response in stress tolerance. So this is going to ensure plants to produce polyamines in controlled way as desired without disrupting normal growth conditions or metabolic pathways. Thank you.